Bismillahir Rahmanir Rahim. In the name of Allah, the most merciful and the most beneficent, countless salutation on Nabi Pak Sallallahu Alaihi Wasallam. So today we will discuss about protein purification and different procedures how to purify the protein. So Aman Safir, please represent your class. Aman Safir, do we have Aman Safir with us? Okay, GR, we have GR. I don't know, I don't remember the name. Anyone else want to represent the class? Please raise your hand. Everyone has to follow the instructions regarding the class that if you have any question, you can raise your hand. No one is allowed to unmute their mic, and if instructor will ask any question from anyone, he or she has to respond, and if he or she will not respond, instructor will remove him or her from the class. So in last lectures we discussed about X-ray crystallography and NMR and different approaches of computational drug designing and structure prediction. Today we will discuss about protein purification. So please represent your class. We have to start the lecture or we have to wait 1 more minute for the other participants. Anyone please? Aman Safir, Abbas Malik, Abbas Malik, 16761. Abbas Malik, do we have to start our lecture or we have to wait for 1 or 2 more minutes for other participants?
वेट कर लें थोड़ी.
Okay, we will
अभी
wait
15
for,
participants.
okay, we will wait for 1 more minute. If anyone has any questions from last lectures, please raise your hand and ask the question. If anyone has any question. Okay, so we will wait for 1 minute. Till that we will discuss about the figure we have on the first slide, that after protein purification we have a figure that is in representing the three-dimensional structure of TAAR6 protein that was predicted with the help of in silico approaches including homology modeling, ab initio, and threading. So in this figure we have three colors: yellow, brown, or maybe red it is, and green color. So these three different colors just representing three different regions of the protein, as yellow color is representing the extracellular region of the protein, that this portion of the protein is from the outside of the cell. And then we have this, in actual it is a transmembrane protein that just moves 7 times from the membrane. If you guys just recall the concept of fluid mosaic model, in which we have two bipolar lipid layers, upside and downside, and then we have intrinsic proteins and extrinsic proteins. If you guys just recall that specific concept, then you can understand this figure very easily, that this protein is also just moves from the membrane, and the brown color is just depicting transmembrane region, that this region is specifically just moving from the cell membrane, and yellow color is just representing the extracellular region, that this region is outside the cell. However, the green color is in the cytoplasm and representing the cytoplasmic region of the protein in the cell. So this protein is the TAR6 protein, that if there is a mutation in this protein, then the patient will have schizophrenia, or some people just pronounce it as schizophrenia. So now we are just moving to our lectures, that pure protein purification. And if you want to do any experimental research on proteins, that this step is very important. Without purified protein you cannot perform the further experiments. If there is contamination, debris, or other you can say other contents of the cell during the purification of the protein, and your protein is not much purified, or minimum 1 milligram per milliliter, then you cannot perform the experiments. And you can say that the reviewer will comment your experiment, or your professor will comment your experiment, you will not have reproducible results, or you will not have reliable results. So the purification of protein is very mandatory step. We are now going to introduce what is in actual proteins. So proteins are the polymers composed of hundreds or even thousands of amino acids linked with the help of peptide bonds, and these bonds just hold the protein and hold the amino acids into one linear chain, and this is also known as a primary structure of that specific protein. So proteins has three different domains that we already discussed in our last lecture. So I'm just going to recall those domains by the names only, and if someone just skip the lectures, he or she can attend the lecture on YouTube about fibrous proteins, globular proteins, and membrane proteins. As I remember, we discussed these three classes of proteins in our physical classroom very extensively. After that, we also have one lecture on this in online classes. So here we have a depiction of fibrous proteins, globular proteins, and membranous proteins. If anyone has any question, please raise your hand. If there will be any distortion in the voice, please report. If there will be some issues with the screen, you guys have to report it. Okay. So here we have fibrous protein in figure A. And you know the fibrous proteins are linear in structure, and usually present and the matrix of the proteins for the stability of the proteins. And usually these proteins are insoluble in water because they have to stable the protein. And if they always just soluble in water, then there will be no stability in the protein. After that, in figure B, there we have globular proteins. That is, myoglobin is the very common example of globular protein, that is roughly spherical in shape. And not all the globular proteins are spherical in shape. However, more than 60 to 70% globular proteins are in spherical in shape, and these proteins can easily soluble in water. These are hydrophobic amino acids are inside the chain, and hydrophilic amino acid of this protein is outside the spherical structure of that globular proteins. Then we have membranous protein, and short before we just discuss about a membranous protein, that was TAR6. And usually membranous proteins has long helical structures because it has to move from the bilipid layer of cell membrane. So here मोहम्मद अफशान जी, बेटा क्या क्वेश्चन है आपका?
अस्सलाम वालेकुम सर।
वालेकुम सलाम।
सर मैंने ये पूछना है कि जैसे ग्लोबुलर प्रोटीन्स यहाँ पे लिखा हुआ है कि ये वाटर सोल्यूबल हैं और हाइड्रोफिलिक्स हैं,
जी
तो
जी।
सर ये अब जैसे एंजाइम्स भी तो ग्लोबुलर प्रोटीन्स में आते हैं, तो उनको तो फिर डिसॉल्व हो जाना चाहिए था क्योंकि वो वाटर सोल्यूबल है।
नहीं बेटा, वो वाटर सॉल्युबल किस कंडीशन में होते हैं? जब वो बाहर आ जाते हैं। इसीलिए इनकी जो अपर साइड है वो हाइड्रोफिलिक होती है, जिसको वाटर हीटिंग कहते हैं। हाइड्रो मीन वाटर, फिलिक मतलब हीटिंग और प्रोटेक्टिंग। तो वो जो बाहर वाली उसकी सतह है, वो अंदर वाली सतह को प्रोटेक्ट करती है, इसीलिए इसकी हाइड्रोफिलिक साइड बाहर पे होती है।
सर हाइड्रोफिलिक का मतलब वाटर अट्रैक्टिंग नहीं होता?
हाइड्रोफोबिक, यस, वाटर अट्रैक्टिंग है ना। तो ये बाई साइड पे है, ये सॉल्युबल हो जाएगी। एंजाइम्स हर बार बेटा सॉल्युबल नहीं होंगे, वो अपना फंक्शन परफॉर्म करने के बाद सॉल्युबल होंगे। अगर वो हर बार
सर ये
सॉल्युबल
थोड़ा
हो
सा
गए
कंफ्यूज
एंजाइम,
है।
मैं आपको दोबारा बताता हूँ। अगर एंजाइम्स हर बार सॉल्युबल हो गए तो वो स्पेसिफिक फंक्शन कैसे परफॉर्म करेंगे? और उनके लिए और बहुत सारी जो इंटरेक्टिंग प्रोटीन्स और इंटरेक्टिंग अदर कंपाउंड्स होते हैं, जो को उनको हेल्प करते हैं मैकेनिज्म में एक स्पेसिफिक फंक्शन परफॉर्म करने के लिए। अगर आप सिस्टम बायोलॉजी में देखें तो कोई भी एक प्रोटीन तमाम फंक्शन परफॉर्म नहीं करती। पूरा मैकेनिज्म बहुत सारी थाउजेंड्स ऑफ प्रोटीन्स और एंजाइम्स की मदद से एक स्पेसिफिक मैकेनिज्म को रन करता है या एक स्पेसिफिक फंक्शन परफॉर्म करता है। तो ये एक दूसरे को सपोर्ट करते हैं।
ठीक है।
So now we were discussing about membrane protein, and membrane protein, you know that it just moves from the cell membrane and bilipid layer of the membrane, that is the cell membrane. And hydrophobic amino acid side chains present outside play role in the cellular transport and often water soluble, but soluble in detergent, often water insoluble but soluble in detergents. So these proteins usually have their binding domain in cytosolic region, or sometimes in cytoplasmic region or outside the cell. The portion that just moves from the membrane usually don't have the binding domain. So these proteins are very important for the movement of movement of compounds from outside the cell to the inside to the cell. So these are these ions and proteins and compounds just depends on these membranous proteins. So hydrophobic amino acids, thank you Afshan for reminding me. I was just after during explaining the question, I just mix up the concept of hydrophobic and hydrophilic. You was right, hydrophobic is water hating, water phobia, phobic from phobic, and hydrophilic is water loving. And globular proteins, upper side is hydrophilic, however lower side, inner side of membranous protein is hydrophilic. And often they are in water insoluble membranous proteins, and soluble in different detergents, because if it will not dissolve in detergents, then you cannot break the cell, and if you cannot break the cell, you cannot extract the protein. So organization of proteins, then you guys know about that primary, secondary, tertiary, and quaternary structure of the proteins. And now we have quinary structure. As I remember, we already discussed it in our class about quinary structure. Primary structure is the stabilized linear amino acid chain of protein is known as primary structure of the protein. And secondary structures usually have alpha lysis, beta sheets, motifs, domains, turns, or other different contents. And these beta secondary structure also have covalent bond, disulfide bond, peptide bond, and hydrogen bonding, electrostatic forces, hydrophobic interaction, van der Waals forces. These types of interactions and forces it has to hold the secondary structure. And then the secondary structure moves and generates a complex three-dimensional structure, is known as tertiary structure. And after that we have quaternary structure, that is consist of more than one polypeptide chain, and work and function properly. Some some proteins just work in dimers, some proteins just work in trimers, some in tetramers. So these all are in this organization of protein. So after that we have the example of primary, secondary, and tertiary structures here: human insulin, myoglobin, and chymotrypsin. So the question is purification of protein. So after recalling the basic concepts of protein, now we are on purification of protein. स्लाइड्स नजर आ रही हैं आपको? Someone please respond. Anyone please respond. Slides नजर आ रही हैं? अलीना खलील, थैंक यू महविश, थैंक यू बेटा जी। बताएं महविश फरियाद।
यस सर आ रही हैं।
ओके, थैंक यू सो मच। So suppose if you are going to here we try to understand the concept first. If let's say you have some bacteria or some cell or some tissues from where you can just take the protein. So do you guys remember the concept of recombinant technology and producing the protein from bacteria? Do you have this concept, or we have to discuss it first? Please someone let me know. Anyone please? जी फातिमा नूर बेटा।
रिकम्बिनेंट डीएनए टेक्नोलॉजी में हमारे पास बेसिकली डिफरेंट डीएनए मॉलिक्यूल्स को ज्वाइन करते हैं टू डिफरेंट स्पीशीज के लिए।
जी नहीं तो ये आप लोगों को पढ़ा हुआ है आप सबने कि इसको मैं रिकॉल करूं?
सर पढ़ा हुआ है, सर पढ़ा हुआ है, 2 सेमेस्टर में पढ़ा हुआ है।
पक्की बात है, रिकॉल करने की जरूरत तो नहीं है?
आई थिंक सो नहीं।
अच्छा ये ना हो बाकी स्टूडेंट्स पहले आपका गिरबान पकड़े बाद में मेरा पकड़े।
नहीं आप कर देते।
अच्छा चलें, मैं इसको थोड़ा सा, हम इसको ना रिवाइज कर लेते हैं 2-3 मिनट ना, रिकॉल हो जाएगा, मेरा भी रिकॉल हो जाएगा, इनका भी। तो recombinant technology जो है, अगर आप, if you guys remember that insulin, production of insulin and these types of protein generation or producing of protein is very important nowadays. So let's see, let's say if you have some bacterias and you want to produce some proteins in the bacteria, so your first step will be the lysis of that bacteria, and then you neutralize the proteases and that proteases that become lysate after that. I hope you guys just remind these terminologies. Proteases will proteins will be the whole part of the proteins in solution, and this step is very important, that if you want all the proteins. Next, if we have to think about the recombinant protein that we created in order to purify it, several different purification methods are available, and every method has its own pros and cons. So if you have charged proteins, we will discuss these all in detail. However, I'm just trying to deliver the concept first, then we will have a look to all these in detail. That if there is a charged proteins and the protein has overall a charge because of excess, if there is so many arginine and asparagine residues in the protein, then the protein will be overall has a charge. Perhaps it can be purified by running it through an ion exchange column or through ion exchange chromatography. For negatively charged proteins, usually the researcher use anion exchange chromatography. However, for positively charged proteins, cation exchange chromatography used by the researcher to just purify the protein. These steps are very important and simple to dissolve the protein in the buffer and then just incubate the protein. Incubate the protein in a resin to just extract or to purify the protein. After that, you have to wash that resin with different buffers, salt buffers, and then you have to elute that bounded proteins with some high salted buffer. First you have to use the low salted buffer, and after that you have to use the high salted buffer to break the ionic interaction that the purified protein just bounded with the resin. So if the protein that was if the protein that purification is in the case if the protein has charge because of asparagine and arginine. However, if the size of the protein is very large or very small, so you guys know that the protein purified because of depends on their charge, on their size, on their nature, and on their isoelectric point. So all these purification techniques utilized here to purify the protein depends on their charge, size, isoelectric point, and other specific you can say properties. So yeah, so when you have different sizes of the protein and you want to purify the protein, exclusion chromatography technique will be applied that can help the researcher to isolate the specific protein on the basis of their size. In that case, dialysis is a technique or approach, you can say, that used to purify the protein, in which the incubation of that specific protein will happen in dialysis bag and stir it while replacing the buffer outside. The target protein and the other larger proteins are retained in the bag, while the smaller proteins that will be not your target protein, because you are targeting the larger protein that is of interest, will be filtered out through the process of diffusion. If you guys remind the concept of diffusion from the chemistry. Size exclusion chromatography similarly separates the smaller proteins out from the bag. However, the larger proteins just be in the bag, and then you have to use HPLC technique to extract that larger protein, and then you can separate or purify your specific target protein that will be enlarged in size. After that we have another one domain, that is protein affinity. So you guys just grabbing the concept or just listening? Do we have Aman Safir? Fatma Noor? No? Okay, thank you. बेटा
Yes
समझ
sir.
आ रही है?
Yes sir, आ रही है.
क्योंकि वो स्लाइड वही वाली नजर आ रही है, प्यूरिफिकेशन ऑफ प्रोटीन्स वाली, क्योंकि बंदा बोर
जी
हो
sir.
जाता है ना एक स्लाइड देख के, तो वो कई मरतबा फोकस जो है वो डाइवर्ट हो जाता है. सो आई एम जस्ट ट्राइंग टू
जी sir.
जस्ट डिलीवर द कंसेप्ट फर्स्ट and then we will discuss it later. Okay,
Okay sir. Yes sir.
so if the lecture will become exhaustive or bit fast, you can raise your hand and let me know. Okay,
Okay sir.
thank you. So we discussed about size dependent protein purification and charge dependent protein purification. After that we have the proteins that have different affinities, and this one is just bit difficult and expensive process to buy the resin for the specific proteins that has specific affinity, as that resins should also have antibodies that will be specifically bind to your the target protein. So you can simply pass your protein through the resin and it will selectively bind to that specific protein that will be the target protein because of that antibodies, and wash it a little bit with buffer so no other proteins are bound and only the target protein can bound, and finally elute it by disrupting the antibody protein interactions. After this one we have substrate proteins, and if the protein is an enzyme with the binding pocket, then you can also immobilize your substrate on a column. I hope in these slides we will have a look to the columns, and if you studied chromatography earlier, you will know about the column or have a sketch in your mind about the column. We were discussing that if you can also immobilize, some columns are mobilized and some columns become immobilized, depends on the nature of the experiment or the protein or the purification technique. So immobilize the substrate on a column and use that for purifying the target protein. Simply pass the protein through that column multiple times so it can bind to specific substrate, while other non-functional proteins can easily be washed away. However, the target protein will just interact or attach with its specific antibody that will be used. G. Afshan. Afshan.
जी सर, मैंने ये बस कहना था कि ये जो अभी हमने टर्म्स डिस्कस किए हैं सर, अगर इनका भी अगर यहाँ पे लिखा होता तो ज़्यादा बेहतर नहीं था?
बेटा आपकी बात बिल्कुल ठीक है और ये नीचे लिखा हुआ है तमाम स्लाइड्स में, तो आई एम जस्ट सेइंग कि मैं आपको भी दिखा देता हूँ। ये आगे 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 आ गई, ये देखें, ये मॉलिक्यूलर साइज वाली आ गई जो हमने डिस्कस की है, ये जेल फिल्ट्रेशन या आयनिक चार्ज वाली आ चुकी है। ठीक है, तो इन डिटेल डिस्कस करने से पहले ये जेल इलेक्ट्रोफोरेसिस भी आ गई है। मैं ये चाह रहा था कि अगर आप एक बार फोकस होके सुन लेते दो दो तीन तीन लाइनें स्पेसिफिक क्रोमेटोग्राफिक टेक्निक के लिए, तो आपके कंसेप्ट थोड़े क्लियर हो जाते नीचे समझने के लिए, और कोई मकसद नहीं था। आप कहते हैं तो मैं छोड़ देता हूँ, इस तरह ही नीचे से पढ़ लेते हैं।
नहीं सर, ठीक है, ऐसे
पक्की
पहले से
बात?
हम अगर थोड़ा डिस्कस कर
हाँ,
लें
वो
तो
इसलिए होता है
बेहतर।
कि जरा क्योंकि कई बार स्टूडेंट वो फिगर्स देखने में महव हो जाता है, तो वो सुन नहीं पाता, कई मतलब वो सुनने में महव हो जाता है, देख नहीं पाता। तो इसलिए मैं इस मौके पर सिर्फ दो दो तीन तीन लाइनें डिस्कस कर रहा हूँ, अगर आप इजाजत दें तो। और आपने इजाजत के टाइम पे म्यूट कर लिया अपने आप को। Anyhow, we were discussing about some protein techniques, and so okay, I forgot where I was in actual. So in we were just discussing about recombinant purification of recombinant proteins. So as you guys know that in recombinant technology we have to add a specific portion of cloned gene to a bacteria, and then bacteria just produce some proteins for us, and we can just purify those proteins with different methods, and this will be our preliminary words in actual. Thank you, Afshan. So now we are on purification of proteins. Why we need to purify the proteins? To identify structural and function properties of the protein. So if you want to know the, or if you want to analyze or evaluate the function of a protein, first you have to purify the protein for wet lab experiments. However, in computational biology you don't need to purify the protein. So your topic is your course is proteomics, so we have to discuss these techniques here. To study the regulation mechanism and interaction level of proteins, we have to purify the proteins. To generate or produce new antibodies for that specific target proteins, we have to purify the proteins. To analyze the structure of protein and how this structure works and how this structure is similar or different from its family proteins, we have to purify the proteins. So where do we get the proteins? So we can extract protein from cells, from tissues, from organisms, from bacteria, from viruses. Depends on the nature of experiment and your expertise, or on from which protein you want to extract the, from which organism you want to extract the protein. It's easy to extract the protein from cells. However, it's bit difficult to extract the protein from tissues and organisms. So there are different steps of purification, protein purification. So first step is the solubilization of protein, in which we have homogenization, centrifugation, and filtration. And we will discuss all these in the later slides where we have these topics. We will not discuss it here. In step 2 we have stabilization of proteins. Step 3 we have isolation of proteins. Some biological materials themselves constitute a clear or nearly clear protein solution suitable for direct application to chromatography column after centrifugation or filtration. So in this figure we have a bacteria BL21, and the specific protein is expressed in that bacteria. After cell lysis and different steps, we want to extract that specific protein that we generate by using recombinant technology. We just produce in that specific bacteria. So here we have homogenization on the laboratory scale. Sonication, it has a sound that just break the cell membrane or other contents of the cell. So here we have different machines, that sonicator, French press, high pressure homogenizer, and these all just used to purify the protein in different steps. Like here we have a freeze thaw, freeze thawing, and high pressure homogenization. We also have that just break down into very small, and then we have bead milling, the specific beads that was produced with these machines. And freezing thawing is also, if you want to freeze it, usually we have liquid nitrogen and minus 170 temperature and minus 80 temperature deep freezers. And for thawing we usually use our hands to thaw the chemicals or proteins, because if we will give more temperature, the proteins will denature. And if the protein is in the frozen form, we just take the vial in our hand and just move our hands, uh, and with the heat of our hands we just thaw the protein. This is known as thawing. Yeah, thaw the protein. So after that we have centrifugation. In centrifugation, we have a machine in which we just put the vial, and the machine just moves so fast in rotation that you usually known as uh, RPM, round per minute, round, I think, RPM, yes, something like that. So So with the help of centrifugation, different isolation processes will perform. Sometimes the proteins just settle down and there we have supernatant of the debris, and sometimes we have the debris in the supernatant in the pellet, and we have protein in the supernatant. Depends on the protein and the type of experiment. So we use this centrifugation machine for this purpose. So we are running of time for this lecture. So we are going to summarize our lecture till this slide. If anyone has any question, please raise your hand and we will continue this lecture in this slides in next lecture. If anyone has any question, please raise your hand. Okay, thank you everyone. We will continue from here in next lecture. Thank you so much for joining.